Hello, so I am Anthony Bonacolta. I am a postdoc in the Keeling Lab and also a member of the ISOP Communications Committee. This is the first of our member spotlights that we're hoping to include in our Revive newsletter. So today I am here with Dr. Um, Julius Lukeš. I think I got that pronunciation correct, who is a recently elected member of the National Academy of Science and also president-elect of ISOP. So thank you for being here.
Pleasure.
First question, why did you decide to study protist?
Yeah. So I have to really go back for young people, very uh, long distance, 40 years plus. When I came to the Charles University in the early 80s, 81, I started. And as students, we were meeting in a pub. And there I heard that parasitology some parasitologist, or I don't know who that was, but parasitology was recommended to me. And so I went there eventually, pretty early in my studies. And and they took me. People were non-political. That was very important. Okay, they did not. So it was kind of island where we did not touch on at that time political things that could bring me in trouble or them. And you felt that the atmosphere is good. And they were interested genuinely in science, and they were good scientists. There was also a tradition of parasitology and protistology in Czechoslovakia before the communists. Giardia was discovered here, and um, some other organisms. So yeah, I stayed, and I'm glad I did.
Yeah. And I understand from some notes we've passed back and forth that you have been involved in ISOP for a while now, and you've had various roles in the organization. How would you say ISOP has kind of shaped your career or your involvement has kind of like helped your career in any way?
Uh, so not that ISOP would be decisive for my career. I would not say that. Uh, I uh, you have to keep in mind that 20 years ago, ISOP was pretty much American-centered organization. It got really international not so long time ago. So for us, it was something distant, and you had to pay a fee. That for a post-socialist country was a lot of money. So all these things are now not really important. I mean, not even for younger guys, not just for me. Anyway, I for me the most important at that point, relatively early at the career, my career was ISEP, the International Society for Evolutionary Protistology, as I organized a meeting in 2000 in České Budějovice or Budweis, where I am actually talking to you from, and. While I had no prior experience with these things, it just panned out very well. We had good speakers, some people who came for the first time to, let's say, form a socialist camp,
Mhm.
and they were kind of surprised that it's not as bad as they thought it was. There were young guys interested, so we really teamed up. It was a very important meeting. And after that, I thought, okay, well, these societies, they mean something. There is something to it if it is done right, and it just so I started following ISOP, and at some point they uh, offered me if I want to be a vice president for 2 years, a long time ago, 2007 till 2009. I had to look it up a little bit. Uh, Uh, I I did not talk much about the business of the society. I still considered myself a kind of observer or junior or careful from small Czech Republic talking into the business of Americans. Okay, but yeah, but in the end. looks like I will have to do that.
Congrats on your election to the National Academy of Sciences. Um, with that position, you'll kind of be like representing protostology on a larger stage and amongst like people who are experts in their own fields of science. How do you plan on using this position to advance protostology?
Yeah, well, that's a tough, tough question, and if I really don't know yet. Okay, uh, the I will enter officially. I mean, we are now um, members, the newly elected guys, but we will be. And I forgot, I never remembered that famous word how, when they take you into the society officially, but that will happen in April. And interesting is that they elected only 24 non-US fellows this year, which is for if you consider all fields of science, it's a ridiculously small number. They can actually appoint 30, but they decided only 24. But Patrick Keeling, and you know him well,
Yep.
and probably most of those who will ever listen to this chat. So he was also elected. So of course, and we did not meet each other ever since because he had to cancel his participation in San Feliu, where we wanted to do that. So, but we have plans to see each other, and we will discuss that, of course, how to let's say sell protists better than so far. PNAS is a little bit also, I think, centered for the classical model organisms, although less than, for example, EMBO journal. So, so let's see. But yeah, I would like to. I think people are elected to this society, and I looked it up relative at a relatively, let's say, late stage of their careers. And I hope I will still be active for some time before I become senile. And in that meantime, I, I want to, yeah, to popularize protest one way or another in on the pages of PNAS.
Yeah. Kind of in line with that, like, would you say there's any significant mentors in your like scientific career that have kind of like um led you here to where you are today, or like significantly influenced your research or your personal life or any of that?
Uh, I think every scientist has to say yes to such a question. Okay, there are of course some geniuses that came out of nowhere from a small village somewhere in the Central Asia. I'm sure there are cases like that, but I'm very normal guy who was fortunate with supervisors and friends and fellows who helped me a lot. I, this age, appreciate it more than, let's say, in the past, maybe. So the really guys' names that I would mention is my supervisor of in Czech, Jiří Vávra. He passed away about 7 years ago uh, at at a decent age. So he really was introducing me to protists with a lot of enthusiasm. He was a microsporidiologist for 65 years or something like that of his life. uh, then I went for a postdoc, my first postdoc already after the Iron Curtain fell, because before that, that was out of question. And I ended up in a lab in Amsterdam, University of Amsterdam, where I was supervised by Rob Bene, molecular biologist who had to study trypanosomes and was not really happy about that. Okay, but then he made a big discovery of RNA editing, almost on a Nobel Prize level, and it was fun. I went there in some kind of really vibrant time, and I learned a lot from those guys. and yeah, and then on the way, there were many people, and as maybe I thought, yeah, I will mention several Canadian guys whom I met in the Canadian Institute for Advanced Research, where Patrick invited me about 15 years ago, and that was kind of very cool, nice experience. So talking to Ford Doolittle and Andrew Roger and John Archibald and Patrick was defining, and they will have a smile and say, "Yeah, sure, Julius is blah blahing about this, but I mean it." So it's a moment to say that.
Yeah. Do you have any advice for early career researchers or anyone just getting into the protostology field?
Well, yeah, I'm I'm aware of the fact that young people don't look that much for advice. I was of that nature as well. Uh, so I would just say my feelings. First of all, if I was young again, I would do the same. So I don't regret. And that has no connection to accolades or prizes. It's really the passion for the organisms. I believe that there will be big discoveries in protists because they are the hidden 70% of eukaryotic diversity on this planet. Uh, while people study every amino acid in every protein in a mouse or Caenorhabditis, or which cell number 692 divides when, okay, there are and you have to do every control just to get their attention, and you have to uh, have everything working, and then maybe they will kind of. But when you study some obscure group of protists that only two labs study, they will actually appreciate it. Everybody will be happy and saying, "Hey, this is good, yeah, that you study it." I mean, of course, the challenge is to get money for it, but the atmosphere is changing in Europe to the better. So, for example, at the European Molecular Biology Laboratory (MBL)
Mhm.
in Heidelberg, this was kind of ignored in some protists. Okay, it's model organisms. We dig deeper. We go for the interaction between proteins and what details. Okay, small RNAs, what they are doing, and so on. Developmental stages. Now they admit, okay, maybe it's time that we look around, that we sample again, that we go after 50 years. Let's go into the field, and the massive sequencing is bringing it to them anyway. Okay,
Mhm.
so I hope there will be more support on the level of the European Research Council, of other national funding bodies for the research on obscure organisms that are not necessarily human parasites. Okay, we don't have to just put everywhere that we will figure out a drug, because nobody believes that these days, fortunately.
Mhm.
Uh, so I think it's a good field to study, and it's fun.
Yeah, yeah. Well, I I agree completely. With that in mind, are there any current or future projects that you're excited about?
Uh, yeah, so my I think my disadvantage and advantage at the same time is that I do too many things. So and I'll get excited about something very quickly and stray away from what I should, let's say, go deeper in. But in the end, I don't regret this risky behavior in
Yeah.
this to some extent. So we do a bunch of things. We decided to study more marine protists that are not studied, and do things like lopid and really solid proteomic studies, uh, try functional studies by knocking down genes, let's say, assembling mitochondrial ribosomes
Hmm.
in organisms where nobody cared, but they are, of course, not boring. Uh, then we have found that transfer RNA can be changed in protists, and now we see it in other organisms in some way. This is, by the way, nice, and I will mention it very quickly. That most of my career spent with trypanosomes, and the paradigm was that all the oddities of trypanosomes are just because they have this complex life cycle. They are just early branch. Remnants of the RNA world, RNA editing, just permanently escaping the immune response, and so on. So that's why they are so weird. And no, all the features that they have are there at extreme. But when you look carefully, those phenomena are everywhere. So they were really eye openers in a way for many substantial discoveries. And this was with ciliates and with other protists. There are Nobel Prizes that actually are not really associated that much with protists, but they should be. So yeah, that's what we are doing.
Great. So I guess I'll end with one last question, and it comes based off some information you said earlier. Any advice for scientists like doing science during complicated political times? Because like you mentioned, a lot of your work was during the fall of the Iron Curtain and all of that work. And
Uh-huh, uh-huh.
being from the US, kind of relevant to me too. So any advice there?
Okay, well, I think that's a new type of question,
Yeah.
really. uh, yep. When I came from a family that has nothing to do with science, okay, and but when I got excited in biology, my father told me, "Well, that's a good field because it is apolitical. I mean, you cannot uh, make politics out of how a tissue is behaving or how blood pressure is controlled. I mean, it can be actually influenced by politics, but okay, so blood pressure was not a good case, but how to breed zebra fish in an aquarium, things like that, okay,
Yeah.
or how to do genetics of plants, little politics there. The communists were able to install politics even in that. And so nothing is totally apolitical, but it is an escape.
Mhm.
And so we should not be that much concerned. We can escape those things, okay, better than others.
Yeah.
Uh, of course, we depend on money that is generated by someone else, uh, and uh, and that's when politics comes into it. Uh, so, not that I have any insight. I certainly, uh, I certainly kept my opinion,
Hmm.
or I tried, even if it was risky. Uh, I thought to hell with it, but I will not do what I am told by the big guys because the big guys were stupid. Okay, so but of course you can do this only to a certain level because
Yeah.
otherwise the system will roll over you, not even noticing the bump. So well, and another thing maybe I would recommend is being something like a moving target. Okay, that you do one or two things, so that if the funding dries in one way, you still have a choice. You can, yeah. I never went really in one direction, putting all my eggs in one basket. That was not my
Yeah.
strategy. Uh, so in the less certain times, maybe I would advise something along those lines.
That's great. Yeah, that's super helpful actually. Well, I think that pretty much covers it. I really appreciate your time,
Yeah,
and
pleasure.
yeah, thanks for being the first of our member spotlights. Appreciate it.
Anthony, thanks for talking to me. Have
Thank
a
you.
have a good one.
You too.